Thank you for coming today for the North Carolina Composting Council Lunch and Learn webinar about unlocking the secrets of soil with Dr. Carlos Goller. And I'm so excited to learn more about the DNA sequencing and technology with composting. So if you don't mind, Dr. Goller, I'm going to share about the next few slides quickly, a little bit about North
Absolutely.
Carolina Composting Council, and then we will dive right in. So, since 2007, the North Carolina Composting Council has been an authorized affiliate organization of the U.S. Composting Council. The North Carolina Composting Council promotes the programs and activities of the National U.S. Composting Council. And it is our mission to advance organics recycling and compost manufacturing, to promote compost use to enhance soils, and to provide economic and environmental benefits for our members and society. Next slide. So, with membership to the U.S. Composting Council, if you are a member and you live in North Carolina, then you would automatically become a member of the North Carolina Composting Council as well. And your membership enables us to encourage and guide research, to promote best composting practices, to educate professionals and the public, to enhance product quality and markets. And then in return, members also receive discounts to annual conferences, webinars, and other events. Next slide. And so the North Carolina Composting Council is our own 501c3 nonprofit organization. We are a grassroots volunteer-run group dedicated to the development, expansion, and promotion of composting and compost use in both domestic and industry settings in North Carolina. Our work is based on sound science, principles of sustainability, and economic viability. All right, and so now we will segue into the Lunch and Learn webinar. I will give a quick introduction of Dr. Carlos Goller, and then I'll pass over the microphone to you. So, Dr. Carlos Goller is a faculty member in the Department of Biological Sciences at NC State University. Since 2012, Dr. Goller has developed courses using sequencing technologies to explore microbial communities and environmental and household samples. Carlos loves collaborations with other classes at NC State and beyond to connect students, technologies, and data sets. By working with undergraduate researchers in the Engineering Sustainable Composting and Outreach Program with the acronym eScoop, Dr. Goller, directed in partnership with Dr. Kakish, Dr. Goller learns about new ways to leverage portable genome sequencing technologies. Carlos, Dr. Goller appreciates engaging with community members as another source of joy and exploration, and we're so happy to have you here with us today. Thank you for sharing your time, Dr. Goller, and I will pass it over to you.
Thank you so much for inviting me. And I really enjoy learning about different environments, learning about composting, and this is something that happened to me in the last couple of years. I became really interested in what can we do with some of the sequencing technologies to learn more about composting as a process. And I'm coming from the microbiology and the technology side of it, but I think there is so much I can learn from you all. So I do have a link to the slides. The link to the slides is go.ncsu.edu/compost SEQ for sequencing. Just in case you want to follow along or if you want it for more references later, uh, the slides are here. And I'm also happy to answer questions. So I'll start with a little introduction. I'll talk about Carlos and composting and how I got into composting, and then a little bit of DNA and sequencing technologies. Uh, I'm particularly fascinated by this new, more accessible technology called nanopore sequencing that seems out of this world, yet is becoming more and more available to us. And then I'll walk you through one of our analysis that we can do with freely available tools, and I need you all to help me interpret. So I'll leave you with some food for thought and some questions about how can we as a community learn more together. So I am a microbiologist by training. I love working in the lab, working with bacteria, growing organisms, incubating, and nowadays back here you can barely see it, but in addition to growing bacteria in the lab, we can take their DNA and sequence it and learn more about which microbes are there and what can they do. And this is one of the instruments I had access to, and now I moved departments. I don't have this particular instrument, but we still have access to nanopore sequencing devices that are becoming more and more uh, accessible, and more approaches are coming up that people can use to sequence a variety of samples. So I was born in Mexico. I majored because I was really stubborn. I majored in biotechnology because I wanted to learn more about this new environment or new research area of sequencing viruses, or learning about Zika and Ebola virus, or learning about infectious diseases. So I majored in biotechnology, and I continued on to microbial genetics work at Emory as a PhD student, and loved working with the Centers for Disease Control, learning more about sustainability. And in the last couple of years, after going to Duke and coming here, my position is mostly teaching focused. So whatever I do, there has to be a training component with undergraduate students helping us learn about our sustainable practices, or how can we make practices more sustainable, and in the process learning about genomics and sequencing. I love speaking Spanish, audiobooks, family, and I do have a blog, and no longer a pet gecko. Now we have a hamster. And I want to learn a little bit about who else is here. So we're going to try this out by using this tool. If you can either scan the QR code, or hopefully go to wooclap.com and enter SEQ25 as your uh, event code. And I'll give you a minute to log in. You can also use the QR code, or uh, even send a text message. So you learned a little bit about me. Now I want to ask you all, who are you? What word describes you? So if you can, hopefully you can vote. Go to WooClap, enter SEQ25 as a code, and let's see what we can share. Two votes already. So educator, outreach, regulator. So we have different people in the audience. And that's fantastic because I also consider myself an educator. I love working with our outreach and extension, and I am learning about what can we do in terms of policy, in terms of education, to learn more about and include more sustainability in the courses we teach. So thank you for doing that. We'll do a couple more polls throughout this. The first one is I want you to think about biotechnology and microbiology. And I'll first ask, what do you think biotechnology is? What's your best guess? So if we look at the word cloud, it looks something like this. And technology to advance biological studies, yes. And when I think about it, it's tools and techniques to learn about the world. And it's constantly changing. Like the fields biotechnology addresses are constantly changing. The areas of research are evolving. And there are numerous connections between disciplines, which brings forth exciting opportunities. And here on the right, you'll see um, a plate with bacteria and fungi from an environmental sample, and you see the variety of different organisms. And with biotechnology, we can use some techniques to learn about the microbial world around us, and that's what really excites me. And several years ago, I was lucky to meet these two fellows here at NC State, Adam Bensley and Matt Ball, in the sustainability program here and in the composting unit. And they took me out and they showed me piles and taught me about waste reduction efforts on campus. And with a summer student, Maureen Haynes, we took some samples from mortality composting units and composting units on campus and learned about what microbes could be found through sequencing approaches in those samples at different time points. And that started this journey, which has evolved over the last couple of years to this program that Olga Kakish and Mahmoud Sharara here at NC State helped merge efforts where we had Mahmoud focusing on waste reduction and does a lot of extension work, and Olga Kakish from engineering that does a lot of mentoring work, creating a mentoring experience for undergraduates to learn about composting and sequencing and biotechnology. So some of the objectives today, I want you to learn about and be able to describe some methods used to to detect microbes present in compost samples without needing to grow them in the lab, which sometimes is challenging, impossible, or unsafe to do. We'll talk about some of the advantages and disadvantages of using DNA sequencing to analyze microbial communities, such as those found in compost. And then we'll end with some interpretation of microbial community data, which sounds overwhelming. It is for me, and you are all the experts who know about your composts and can tell me a little bit about what these samples may or may not have and what may be good microbes to have, for example. So I'll start with part one, which is a from garbage to garden case study on how we prepare samples for sequencing. And this is a student here at the agroecology lab taking some compost from campus and using it for our on-campus gardens. And the first question is, how can we get DNA from soil? And the easy answer is microbes and other organisms, so bacteria, fungi, viruses, small eukaryotic organisms, protists, are found in soil and compost. And we can extract total DNA from the community. This is often called metagenomics or metagenomes, or you may have heard the term microbiome. And this is the collection of DNA from a microbial community. And to get to it, we typically use beads and enzymes to pop open and purify the DNA. And this is the term that you've heard uh, maybe in the news, microbiome or gut microbiome. Well, there are microbiomes in compost. There are microbial communities in compost that depend on various factors like the compost health, the environmental conditions, location. And today we're going to investigate how we go from soil or compost to DNA to making inferences on what microbes could be in that sample based on sequence analysis. So to do this, we typically have a student take and weigh a sample after preserving it. So we either quickly freeze it or put it in a solution to preserve it. We place a small amount of compost, in our case, about 200, 250 milligrams of material in what's called a bead beating tube, which, as the name implies, it's a plastic tube that holds about 2 milliliters of substance and has tiny powder-like beads that, when agitated, help us beat up and beat open the microbes and yeast and get DNA. This is the shake to break system. There are other more gentle approaches, but for the most part, this works to get DNA out of them. And then we bind the DNA to a column or beads to purify and wash away proteins, lipids, other impurities, and collect the DNA. How this happens, there's a really nice video from this company, Kaizen, that I'm going to play because it nicely summarizes what happens.
Microbes are everywhere. These microorganisms and their metabolic activities are essential for this planet. Today, we will look at one of the most abundant and diverse ecosystems on Earth, soil. Soil is a challenging sample to study, since its DNA is based on the quantity and type of soil. Let's have a look. Soil samples collected from the same source have variable microbial load and different organic makeup. After collection, use your sample as soon as possible, as prolonged storage will affect the microbial composition. For later use, store it in Power Protect DNA RNA or put it in the freezer. For small sample with large microbiome load, use the DNeasy Power Soil Pro kit. For low biomass samples, use the DNeasy Power Max Soil kit that features tubes that can handle larger volume. The kit is shipped at room temperature. When you receive it, make sure that you take the solution CD2 out and put it in the fridge in order to prevent precipitation. The rest of the kit can be stored at room temperature. Are you ready for the extraction? Let's start with the lysis. Briefly spin the PowerBead Pro tubes to make sure that the beads have settled down. Use a weighing paper or funnel to transfer the soil and put it into the tube. Make sure to not overfill to prevent inefficient disruption. You can also store the tubes overnight in the freezer. Before disruption, make sure to thaw to room temperature to avoid tube breakage. Adding solution CD1 to your PowerBead Pro tubes would help disperse the soil, dissolve the humic acids, and protect the DNA from degradation. Briefly vortex your sample before I show you our new instrument. This is our new Tissuelizer 3. Place your tubes onto a tube adapter and make sure you balance it with empty tubes. Place the adapters onto the instrument. The tubes are now ready. Disrupt the sample using 25 Hz at 5 minutes, or use one of our pre-installed protocol called Soil DNA. Now, reorient the plate to ensure efficient sample disruption. Since we use 2 empty tubes, we don't need to rotate the second plate. You can now start with the same parameters. After the disruption, centrifuge your tube to pellet the solids. Transfer the supernatant in 2 ml microcentrifuge tubes. Your sample is now lysed and homogenized. Add solution CD2. It contains inhibitor removal technology that would precipitate the contaminants that interfere with your downstream application. Transfer the supernatant into 2 new collection tubes. Add solution CD3. It will selectively bind DNA onto the membrane of the MB spin column. And make sure to not touch the pellet. vortex the tubes for 5 seconds, and then load the lysate mix onto an MB spin column. Centrifuge the tube and discard the flow through 2 times to make sure the lysate has passed through the spin column. After the second centrifugation step, discard the flow through and put the MB spin column onto a new collection tube. Make sure to not splash the flow through on the MB spin. Now, add solution EA to remove proteins and other non-aqueous contaminants. Discard the flow through and put the MB spin column onto the collection tube. Now, add solution C5 to further purify your DNA. Then, repeat the washing step. Discard the flow through and place the MB spin column onto a new tube. Make sure solution C5 is thoroughly removed as ethanol interferes with DNA downstream applications. Place the MB spin column onto an elution tube and put solution C6 at the center of the membrane to ensure complete release of the DNA. The tube is now ready for the last centrifuge step. Discard the MB spin column, and that's it. Your DNA is ready for downstream application. We recommend freezing the DNA as solution C6 doesn't contain EDTA. To concentrate DNA, refer to the troubleshooting guide.
So I played that 5-minute video because that's the entire process. We go from soil, putting it in a tube after weighing, vortexing it to disrupt and lyse open. This is the bead beating method. And then you extract the DNA and purify it from other impurities through a spin column and some washes. And this is fairly simple. We do it in the lab in 20, 30 minutes and can do several samples, as well as there are automated options that we can use robots or automation to do this for us. So we can go from compost samples, and we have in classes, take time points, collect the DNA from these microbial communities, purify it, figure out how much DNA is there using some instruments that use fluorescent dyes or separate the DNA by size. And then we add some adapters to it in a quick 10-minute reaction to then feed it into an instrument that's a USB connected device that can be run on a laptop to sequence your samples. So we can go from environmental samples all the way to taking that DNA and learning through sequencing what microbes may be present. So how can we get DNA from our samples? Going back to that garbage to garden case study, I'll share an example. So here at NC State, we have Howling Cow ice cream, and once you finish a delicious ice cream, you prepare to throw out the empty container and the spoon into the trash. And then you stop so that it go in the compost bin. And when you look at the spoons and cups that Howling Cow uses here on campus, they are, for the most part, biodegradable and BPI certified serviceware. So you search some information about these cups and you decide that you love research and love NC State ice cream and were curious about the composting and what microbes were involved. And this is a true story. Students on campus convinced me to do this. And we asked the question, what happens in the compost and what microbes are involved? And I want to explain some of the steps we use to identify the microbes in that compost without the need to grow them in the lab. So we begin by visiting the compost facilities, taking a sample, maybe of the partially degraded cup, and compost around it, and ask the question, what microbes are helping break down this cup into the organic matter? So you ask a friend from the biotechnology program to help you figure this out. And we begin by extracting the DNA as we did it, with that video you watched, using bead beating to slice open the bacteria in the sample, separate it from the soil or compost, and purify the DNA. And the next component is we can use an instrument, and this is one of several instruments. There are other technologies, but this happens to be portable. Here's one that's USB, and here's an all-in-one. Called Oxford Nanopore Technologies, that allow us to sequence DNA to figure out what's there. So the next video I'm going to play is 2 minutes. It doesn't have a voiceover, but it walks you through what this technology can do. So that was a fairly promotional video, but it really highlights the piece of equipment that can attach to a computer. It has a flow cell. The flow cell is where you introduce your DNA that has been prepared with adapters so that they can go through the pores in that flow cell. Think about it as a cartridge. And then what happens is the pores in that are nanoscale pores in that flow cell. Your DNA has an adapter that's caught by these tethers on the flow cell, and the enzyme adapter that you added opens up the double-stranded DNA and passes one strand through the pore. And as it goes through, you get disruptions in the current that are measured by the sensitive device, and then using machine learning algorithms, we match these patterns to A's, G's, C's, and T's to learn what sequences are in our DNA. Why is this important? Because we can use this technology to do some of this, which is my next video, which is taking these DNA sequences and learning what, by matching them to databases, what can or what microbes are in this sample in our case. So now going back to our diagram, we can quantify our DNA, add adapters to it, sequence it, and begin analyzing this. There are kits available that are specifically created to sequence either all DNA present or specifically target bacterial identification genes or genes that we can use, sequences that we can use to identify bacteria, or sequences that we can use for for fungi profiling. And then we can do several samples at once by adding different adapters and sequencing them in the same batch. This is often called barcoding. And we can ask questions like, how do these organisms contribute to the degradation of the compostable cups? Do they vary over time? Do the abundances change over time? So what does the data look like? I'll give you a live demo of some data collected not from a cup, but from a compost on campus. And I'll ask you these three questions. What can you interpret? What do you think this means? And why is this important? So you may buy a kit or buy an flow cell and sequence your DNA, and then you'll end up with a whole bunch of files, up to terabytes of files. And for example, we can get uploaded to a website. In this case, I'm using the Chan Zuckerberg, so Facebook initiative, infectious disease platform. They hired a whole bunch of bioinformatics experts and created a platform for people around the world to be able to quickly analyze environmental and health related samples to learn what microbes are there. So you upload your sample. In this case, it had 297,000 sequences, and uh, you can analyze this within 10 minutes of uploading. What does the data look like? It gives you an interactive tree that says we got this number of sequences and base pairs. The bigger the circle, the more of those sequences were matched to a database and correspond to bacteria. Then we have more that match Pseudomonas. And you can open up each of these branches to learn more about the family or the genus or even species level of the microbes found in this sample. This is an interactive tree. So, for example, you could open up branches and check. The statistics it gives you are a number of bases that match. So you can add and change the thresholds. And you can also present this as a table. For example, you can ask for the common names. You can ask for just viruses in that sample. And then it will pop up some links to the National Bioinformatics database, PubMed, and Google Scholar searches for this virus, movie virus. If you wanted, you could also look at uncategorized sequences. You could set thresholds depending on different percent matches, or you could add flags and say, find everything that's a potential pathogen. And mind you, this is one that is more for infectious disease work, but I can sort by how abundant it was. And then it can show me the DNA and what parts of the chromosome of this organism were matched by the sequences in our sample. And you get some coverage and um, fidelity of matches. And you can download all of this as either spreadsheets or an entire folder full of results. So, for example, you could say, I am just interested in bacteria that could be pathogens. Oh, we found one bacterial species, it's E. coli, a strain that could be a pathogen, and it gives you the information. This is one of several tools. This tool has some options to compare samples. They are still working on, for nanopore sequencing, the tools to compare in real time multiple samples from, for example, different compost units. So if I were to ask you, what microbes do you think we will find in your compost? What do you think we'll find and why? And it may be names right now, or you may have some idea of, oh, there may be a fungi that is visible, and I would expect to see an abundance of that. Or at higher temperatures, you may see uh, uh, less diversity of microbes, but the ones that are there are highly adapted or able to withstand higher temperatures. So we can do a lot of investigating with programs like this. There are other programs that I would also recommend. Um, and this one allows you to keep your data private or upload and share it with others. We've done some experiments with plant growth chambers, and the analysis is pretty robust. So people have, or students have taken and downloaded the results in tables and then plotted them on their own. So some things I want to emphasize are there are advantages to the use of metagenomics, the approach that I just shared with you. One advantage is you can sequence basically everything in your, you can sequence all your DNA that you get out of your soil sample or compost sample, and based on matches to database, get an idea of what genes or sequences are there to learn not only what microbes are present, but what could these microbes be doing. You can also, if you have not that much DNA, you could amplify what we call reference genes, those bacterial 16S genes or fungal ITS genes, to learn about the diversity of, for example, fungi in a sample. There are also some primers for or genes that can be targeted for specific viruses and archaea or even microeukaryotes to do what's called barcoding or meta barcoding and learn about specific types of organisms in your sample. And this is a little more flexible because you don't need as much DNA. Many times when you sequence all the DNA in a sample, most of it cannot be matched to databases to give you a defined, I think this is E. coli result. And last, there are numerous kits and instruments to sequence DNA. Both Nanopore is capable of sequencing very long strands of DNA. Illumina is really robust and capable of sequencing much more throughput but shorter fragments of DNA. And now there are numerous services that one can use, and for $100 more or less, you can sequence your sample and get a report of what is likely there. So these are three advantages of the use of metagenomics. You can sequence everything, all your DNA. You can target specific organisms or classes of organisms. And there are numerous kits and services available for learning what is in your sample. There are also several web-based free analysis platforms. The Chan Zuckerberg ID system is one. K-Base is the Department of Energy tool that has several useful tools. There are some company-specific ones like Aladdin. There are some more medical-focused ones like BVBRC. But there are numerous web-based free analysis platforms available to take your sequence data and learn more, mine for certain organisms to learn who's there, or learn about the genetic potential. What enzymes could be present? What could the microbes in your sample be doing? There are some limitations of metagenomics. One is you're basing everything on comparisons to databases that may not have sequences you find in your sample. For example, there may be a lot of unknowns in your DNA because your DNA was not a good match to something in the database, or was ambiguous and was not a match that could give you a certain taxonomic classification. So many times I get people asking me, well, 80% of my total DNA was unclassified. And I said, well, that's really good. You classified 20% of your DNA. Uh, and that happens when you're sequencing total DNA. When you're using 16S, a bacterial gene marker, most of it matches databases because you're targeting databases that specific organisms and databases that have been fine-tuned and developed over the years. So keep in mind that all of this is comparison to databases of the DNA sequences you have and what is available in the databases. Not everything can be specifically or accurately classified. Some sequences are shared among microbes, so they don't provide useful information. They may be classified, but they just tell you this is a gene that's found in all bacteria. Or this is a typical virus capsid gene that several classes or families of viruses have. And you only know what DNA was there. Though you can also sequence the RNA, the messenger here, to see what is actively leading to protein production. Right now, we just talked about sequencing the DNA, but nowadays you can also sequence the RNA fairly easily, also with services, to learn about what is metabolically active. I'll also say that people are working on some really neat machine learning models to determine what looks like old DNA and what looks like new DNA based on this pattern of the current of these bases as they go through the nanopore. So people have been, Laura Urban and others have been creating algorithms to try to detect and differentiate E. coli DNA from dead E. coli and E. coli DNA from live E. coli using machine learning algorithms. But for the most part, these approaches tell you what DNA was there or is there. We're not sure if the organism is still alive or the virus is still in an active form. And there are variations in protocols, so many out there that it makes comparisons of studies difficult. Though people like Nefelli for the NIH, National Institutes of Health, and K-Base for the Department of Energy have been trying to standardize protocols for how we analyze metagenomes so that we can compare across samples. So keep in mind that you are comparing your sequences to databases that may or may not have all the organisms in your sample. Not everything can be accurately classified or specifically classified. If you're sequencing the DNA, we're only looking at what was there or is there. We're not sure what is active. Though you can sequence the RNA, it takes a little more uh, on the lab side. And there are so many variations of protocols that often comparisons between studies may be challenging. So as we wrap up and have some time for questions, my challenge for you is if we analyze microbial community composition and how do we connect this information to environmental conditions? The power of these approaches and techniques is not only to sequence everything and match it to a database, but to make connections with the rich metadata, like rich soil and rich compost. What other parameters like pH, temperature, conditions of your compost, different things you add to your compost, what helps us learn about the community of microbes and what makes this timestamp or the snapshot we get from a metagenomic analysis of the microbial composition, what makes it useful is connecting it back to that rich metadata, the environmental conditions, or how you perturbed the compost. For example, adding more of this or taking away this. So these are some of the challenges when it comes to analyzing metagenomic information. Keep in mind how your unique samples make the analysis even more powerful, because maybe you can compare different different conditions or different sets of conditions and how they affect the microbial community composition. So some takeaways: the microbial communities in soils and compost are truly dynamic, varied, and now much more accessible through sequencing. People can take time points and learn about the development of microbial communities, the changes in compositions of these communities, and even the abundance and genomes of very rare microbes in those composts. I did not talk about other approaches that we're looking into. A good friend uses something called Phase Genomics is his company, but he does deep sequencing of a single microbial community using clever tricks to lock in place the DNA within the microbes to get an idea of which DNA corresponds to which microbe, and if there was sharing of DNA between microbes, and get a better idea of the complete genomes, not just pieces, but bigger puzzle pieces to learn about the complete genomes of the microbes in those communities. Obtaining DNA from soil and compost samples can be done easily. However, the quality of the DNA and the concentrations may vary, and humic acid and other inhibitors of downstream steps are a challenge when it comes to soil and compost samples. And depending on the approach or the kit you use, inhibitor removal can be done. And sequencing of microbial communities is now almost routine. Microbiome and microbiome and metagenomics kits and services are available through companies like GeneSeek or SeekCoast, and Zymo and Kaigen have kits available. You can sequence everything, all the DNA there, as much as you can recover, or sequence specific marker genes to learn about microbial communities, to learn about bacteria specifically, or fungi. And there are short read sequencing technologies like Illumina, routinely performed by sequencing centers and core services, and long read sequencing like the nanopore sequencer that I showed, or PacBio, Pacific Biosciences, are becoming more available through services and teaching labs. And which microbes will enhance composting of difficult material remains an exciting area of research. We may know who's there. We're still learning what are they doing and how can this improve or benefit composting processes. So with that, I will stop here and ask you all, what would you like to see next, or what questions do you have? And feel free to put them in the chat. So one question, how do sequencing results translate into actionable changes for compost management or soil amendment practices? And that's a tough one and important question. I think there's now information about certain microbes or classes of microbes that can benefit or accelerate certain processes, and depending on your soil's physical and chemical properties, you may want more or less of some microbes. So this information can tell you almost or help you gauge what would be useful in terms of amendments to your physical and chemical properties of the soil that would then benefit your microbes. Can DNA analysis detect pathogens or invasive species in compost material? And how reliable is it for quality control? Yeah, so as CZID showed, you can detect known pathogens at a very sensitive level, so 1-2 sequences, and you can then add thresholds for how much match you want to the sequences of pathogens, and it could be used for quality control. When it comes to invasive species, plants are a challenge, and I've been thinking about how can we detect. In my case, I'm interested in wisteria. How can we detect invasive wisteria? And there are not that many biomarkers or sequences we can look for that will tell us this is invasive wisteria or this is native wisteria. Is the role for DNA sequencing in verifying the ecological benefits of compost application? Is there a role for DNA sequencing in verifying the ecological benefits of compost application, carbon sequestration, soil biodiversity, etc.? I think definitely for soil biodiversity and carbon sequestration, nitrogen fixing, people doing plant microbe interactions and the rhizosphere, the microbial root communities know a lot about which bacterial signatures or communities help their soil, help their plants, and I think that can be applied and is, excuse me, being applied to composts. I think we can be more systematic about it and maybe share more of the knowledge of how this is being done, what amendments are being added, what thresholds. That's hard to establish. What is too much of one microbe? It may depend on so many factors like environment, location, sequencing technology used, and all that. What types of sequencing technologies are being used most effectively in soil analysis today? So 16S is amplifying the 16S bacterial gene, and now that is becoming very easy for companies and services to do it in automated ways and bringing down the cost to $20, $50 per sample. Whole genome sequencing is still provides a lot more information, not just who's there, but what could they be doing, and not just limited to bacteria, but also maybe viruses, maybe phage bacterial viruses. The challenge with whole genome sequencing is a lot of that DNA is difficult to classify. So you need what's called higher depth, sequencing depth, to make inferences. So you need to sequence more. How do environmental factors like moistures, temperatures, or feedstock diversity affect microbial populations over time based on sequence data? Or is this still being classified with high sequencing? Or is this still be classified with high sequencing depth? So temperatures do affect the diversity of microbes. It's really interesting because in our hands we've seen just certain microbes at the peak temperatures of the pile or the compost. You see also certain bacterial viruses become more abundant over time. Uh, it's and we've done this based on sequencing data. There are other approaches like taking samples and using mass spec analysis to see who's there, what proteins are being produced. But genomics, hand in hand with some of the protein technologies, can give us lots of information. Great questions, tough questions. Last question. Could this technology be used in regulatory or certification frameworks for compost products in the future? Yes, and one thing I've been pushing for is a class where we actually go in the field and try some of the sequencing in the field or the DNA capture in the field. We have some physics students trying to create a portable homogenizer to use solar power and something more portable than the Kaizen piece of equipment to take the bead beating tubes, collect soil or compost in the field, bead it to start the extraction process as soon as possible to get a more accurate representation of who's there. So I think in the future there will be much more live or real-time sequencing. In clinical settings, people are using it to detect rare genetic disorders in premature babies, and I think the record is a 7-hour turnaround.
That's all such great information, Dr. Goller. Thank you. I actually have one question as well that maybe
Yeah.
we could close on. And you kind of just started maybe into that, but what do you see as the next big breakthrough in compost and soil microbiome research?
Oh, so I think we are getting better at the sequencing and figuring out who's there. I think that what are they doing is still a challenge. And I think now it's becoming easier to uh, get the messenger, the RNA, and learn what's being produced in the compost to get an idea of what enzymes are key. And that tied in or paired with proteomics is really powerful.
It's fascinating and exciting to see that there's a lot more to uncover.
Yeah, and I think it's a great students learn some techniques, and I'm really interested in how we can think about sustainable practices and use of the technology to improve sustainable practices.
That's awesome. If you don't mind, can you screen share the presentation one more time? I'm just going to make sure that we share our contact information with everybody and just
Yes.
close out. But thank you so much for your time today and for everybody watching and participating. We greatly appreciate it. And just a reminder, this is a North Carolina Composting Council Lunch and Learn webinar. We will post them onto the North Carolina Composting Council YouTube page. And I think, Dr. Goller, do you mind going to the very end? I think it's the contact slide and staying connected maybe with North Carolina Composting.
Got it.
So I'll share some of our upcoming events. If you are watching this live, we do have an in-person Sustainable Spirits event in Charlotte on October 16th from 5:30 to 7:30 at the Innovation Barn. And I'll share a QR code pretty soon as well with links to everything that I'm talking about. But I already mentioned our YouTube channel with the webinars. We also have our grant program open right now. So through October 20th, 2025, we welcome applications to apply for composting projects that have a public benefit in the state of North Carolina. And then also I mentioned we are a grassroots member-based organization. So we do encourage membership either to the U.S. Composting Council. There's also other levels on the state membership options. And again, this QR code will link you to all of the active events and other programs that we have open right now. We just encourage you also to stay in touch with us, whether it's volunteering on a committee or at an event or just subscribing to our newsletter. Dr. Goller, if you don't mind going to the next slide, this is a direct QR code to our newsletter and more importantly, to the contact information for Dr. Goller and myself, Catherine Polk with the North Carolina Composting Council. So again, thank you so much for sharing your expertise and experiences with us. We're just very grateful, Dr. Goller. And also, I just want to do one more slide to the next one, if you don't mind. Also, big thank you to the sponsors of the North Carolina Composting Council who enable us to host these free webinars for your educational opportunities. So thank you, Sustainable Generation, Vermeer Allroads, Smith and Gardner Engineers, SCS Engineers, McGill Environmental, Denali. We appreciate your support, and I hope that you all enjoyed the webinar. So thank you very much.
Thank you.
Have a great day.